Our goal in undertaking this work was twofold. First was to understand the mechanisms at molecular and genomic level that were associated with recognition and response to the grafted tissue. And secondly, to use this information to develop markers and assays which would help to predict outcome and our intervention, and then to combine those two to demonstrate novel potential targets for intervention. The process, as you heard described so eloquently this morning by Dr. Lemon, has three components. The first is tests of analytical performance. Are these tests valid, reproducible, and so on? Number two, of clinical performance. When we apply them in the clinic, do they give us the information which will change our practice? And three, economic performance. Let's assume we have a test. Is it now economically viable within the health system where we operate? So going to the beginning, the challenge for us was to take the massive genomic information and the massive proteomic information, to analyze it over time, and to distill this into a series of markers which would predict, in the first instance, rejection of the heart and rejection of the kidney following transplantation. We were successful. You see some of the publications there in a variety of important journals, and these led then to the next steps in the process. The first question now is reproducibility and clinical importance. We must move through a series of phases of discovery, of internal validation, and of external validation. And Bruce will close with some of those external validation programs. But here we need to know, first of all, whether the genes in the donor heart and tissue actually predict outcomes, whether the genes and proteins in the blood diagnose the outcomes, and whether the proteins in the blood diagnose the chronic injury to the graft, because we can have two events, one that occurs early called acute rejection, one that occurs late. How do we put all this massive information into a simple diagram for demonstration? We use what we call a receiver-operator curve, which are shown here. And the receiver-operator curve measures sensitivity against 1 minus specificity for any test. The ideal test would move up the vertical axis and across the top. That's never seen. These are remarkable clinical outcomes for the field in which we operate. And in general, AUCs of over 0.8 are considered very powerful as potential biomarkers. We move on then to ask, can we now begin to combine this information? Because on the one hand, we have genomic information. On the other hand, we have proteomic information. If we were able to bring the two sets together, would it help? And when we look at the fundamental mechanisms, the genomic information tells us about events which occur within the lymphocytes, the T cell, about the recognition mechanisms, the signaling through JAK-STAT pathways, gamma interferon and others. Whereas the proteins tell us about the inflammatory cascade, complement, coagulation systems and others. So we can see here when we combine genomics and proteomics, and we look at some of the sensitivity, specificity, and area under the curve from receiver operating curves, we actually have an improvement in the predictive capacity of these assays, which is exactly what we would hope for, because they look at the question from two slightly different sides. We can now ask really perhaps the most important question. The information that I've shown you comes from our own discovery process and internal validation process. But if we take these markers and we now apply them to data sets which are completely different, for example, large data sets from the United States, do they concur? Do we find roughly similar outcomes? And I must tell you, just for information, that the way we practice transplantation and the drugs we use and the way we use them in Canada are not quite the same as those in the United States. So that this is a big hurdle. But here we show that when we take this data and we apply it, whether with the genomics or proteomics, all combined, in fact, on the right-hand side of your screen, you see that we still get very good performance against a data set and biological series of samples which are treated even in different ways from those that we treat in Canada. So it looks as though, so far, These are accurate, reproducible, and robust because they stand the test of comparison. Now, there are a variety of ways that we can use this information or combine this information. And one of the ways is by using a decision tree process. And a decision tree process says, if A occurs, and we have two outcomes, given one, if then B occurs, does it lead us to a better decision? So we can start with a very small number of proteins. And then, if those are present, we can add to them a small number of gene changes, gene expression changes. And if those are present, we have a very good likelihood, we believe, that we will identify rejection. And in this 2-by-2 probability table, you see outlined or highlighted in yellow that this is in fact the case. The true event rate and the negative event rate are extremely high, and in the margins you will see the marginal probabilities for outcome. So far as we have been able to explore these, it looks as though the path is solid and we are in fact moving in the right direction. But so far we have not met the major test. The major test is coming now, which is our international validation study. As you can see, this involves a large number of sites across areas of the world which use very, very different approaches to transplantation. The enrollment of more than 500 patients has now been going on for a couple of years. We're approaching the end. We have a very large number of samples for genomic and proteomic analysis, as you will see here. We will be using now a slightly different proteomic technique, multiple reaction monitoring, which is more precise and specific than our original approach of iTrack, but the gene expression will be measured in the same way. So our hope and our goal is to see the same biomarkers that we discovered internally and validated and cross-referenced externally, now replicated in this large study. When we develop the biomarkers to our satisfaction, we have to have a very simple translational approach that will enable them to be used in the clinic. Now, this is a very daunting task for many biomarkers, but we're fortunate that there are techniques which are very new techniques which will be applied directly to this. And one of the most important is a technique called Luminex, which uses a small series of microbeads which fluoresce with different degrees of intensity and different colors, and to which various markers can be bound. So we can use this approach for the assay development and testing of our markers. And here we show you from our internal analysis, using only a very small series of our genomic markers, that this assay looks as though it will provide the platform we require to translate this to the clinic. Why is this important? Because this particular kind of assay is used in virtually every transplant program across North America and most of those outside North America, so the translation should be simple. We have described, or I have described, I hope, two of the important tests. The analytical performance of the assays we hope to develop and the clinical performance of those assays. But now we must meet the third very important test that you heard from the first speaker, Dr. Levin. And this is the economic validity or economic performance of the assay. And I'll hand back to Bruce, who will just take you through some of the work that's been done, which is quite extensive.
I think Paul and I are brothers by now. But just on this graphic is sort of a fulcrum for health economists for years and years. And we have the good fortune of working with Carlo Mara, the brighter, younger brother of Marco. And Carlo has helped us with our appraisal of the impact of tests, for acute or chronic rejection, diagnostic or predictive, in the transplant context. And one can see that you're looking at assessing incremental cost-effectiveness ratio and looking at cost of the test and effectiveness of the test. And we've done this, and we've done not only the analysis, but also modeling of the impact in Canadian context and in the US context. And in Canada, just for acute rejection, not chronic rejection, but acute rejection, we're talking about a 5-year savings in Canada, a small market in the world, of 55 million in kidney transplant and 75 million in heart, with very rigorous analysis by our health economics team led by Carlo. And then if you look at this, the way, as again nicely as Dr. Levin indicated, you have to look at this from in terms of what test it is that you're offering, and okay, why are you offering it? What its performance characteristics by standard laboratory context and AUC, and then proposed test cost, the U.S. market value per year, the U.S. healthcare savings per patient over 5 years, the quality of life years impact, and then of course how the regulatory agency views the test, either as a low risk or a high risk test, which will determine with our consultants, with Scientia in the U.S. and with others who are working with us, to develop an entire plan for the migration of these tests, either as laboratory developed tests to the FDA or directly to the FDA, depending upon the situation. And this is just to tell you that for every test that we're developing, we have a strategy that's being developed that is complete from end to end in this respect, that includes health economics, regulatory, and reimbursement. And so to close this, this was an attempt to sort of show you where we're going in the commercialization pathway and implementation pathway in the third phase. So we started with biomarkers in transplantation in 2004. Luminex was identified as a potential clinical platform as early as early 2007. We had the voluntary data submission validation by the FDA in 2009 and discussions with Luminex. We then had an agreement signed with Luminex, a formal agreement in early 2010, and we've been working with Origin to develop the protein assay on a Luminex platform, and a gene assay development, which will be a combination of Luminex platform and HTG chemistry. And then finally, external validation of the decision tree model, pre-IDE submissions. We have intense discussions now about the details of marketing with Luminex, and health economic and market data is being worked on constantly, and then the BIT2 samples are actually on the high-performance platforms as we speak, and we'll have the data by Valentine's Day. So the cornerstones for our success, we think, are, to date, are clinical and research excellence, human life scientists, but especially computation and system scientists. I'm not talking about our system science work today, but we are very proud of Raymond Ng and the team for their recognition globally in Boston last year. And health economics is integral to this, as Dr. Levin indicated, quality focus, no boundaries, and we strongly believe that relationships trump IP. We try to keep legal discussions to a minimum in this not-for-profit organization. So the team involved is the Translation Advisory Committee of really tremendous people in and out of Canada in the public and private sector, across the span of our domains of need, a board of directors similarly, the management team, the operations, the computation team, the trainees, and many clinical collaborators, and a community of partners for cohorts, platforms, computation, resources, science, and health economics, health systems integration, and commercialization. And so that's just a snapshot that Paul and I are trying to give you of the BIT program. And we hope by 2013 we'd be able to report to you that these actually have been validated in a local setting. Thanks.